Yes, now I can hear you.
Yeah, well, rebooting the computer always helps. And this emphasizes my point, which is that I cannot wait for all this COVID, I want to use a strong word, inconvenience to end so that we can meet in person and we can much more easily understand each other and communicate much more effectively. All right, so can you see my screen now?
Yes, we can see your screen.
Excellent. So I'm very honored to be presenting at this workshop dedicated to the memory of Gregor Nicoli. I first met Gregor in 2003. We had had some initial results on non-classical nucleation, and he was interested in the mechanisms of this phenomenon. So he started the theory effort to address non-classical nucleation. And in several years, there was a meeting organized and this book was published, which was co-edited by Dominic. Dominic is now our session chair. The theoretical efforts involved Vasilios Bazios, I can see him in the meeting, Jim Latsko, and Dominic started a large experimental effort dedicated in part to non-classical nucleation. And, we also continue to work in that area. And I think now is the time to make the next step in the studies of non-classical nucleation, which is to move from cataloging different systems which nucleate non-classically, or crystals nucleate non-classically, or to different non-classical behaviors, two-step, three-step, multiple steps. To actually, the next thing is to harness the complexity of crystal nucleation so that we can control nucleation. And the control of crystal nucleation is the holy grail of material science. It was something that was always dreamed of, and it was always thought extremely difficult to achieve. And so before I show you why nucleation control appears so untenable, let me start with introducing you to what crystallization is, because I think that I'm the first talk on crystallization in this meeting. So crystallization may occur everywhere. It can occur in human arteries, or it can occur in industrial pipelines, or in oil tanks. And in many cases, it is benign. And so these are these beautiful selenite, which are just an example of gemstones, or it can be highly undesirable. Cholesterol crystals in the endothelial walls causes heart disease, and kidney stones are extremely painful. So clearly, sometimes we want to promote nucleation, and sometimes we want to suppress it. And then if we look at how crystallization happens, we have to realize that crystallization is divided into two steps. The first one is nucleation, as shown here in this very popular diagram. Nucleation is highlighted in the evolution of crystal populations because it is the step that has to overcome a thermodynamic barrier. And this diagram highlights one additional feature of crystal nucleation growth, and it is that we can have the classical nucleation, which is by assembly of individual monomers. It also lists several non-classical pathways where before a crystal nucleates, we can have amorphous, liquid, or even crystalline particles that facilitate or suppress crystal nucleation. And these are all the different non-classical mechanisms of nucleation. So what we're going to do today is that we will discuss crystallization and nucleation of hematine. I promised something else before that, and it is why nucleation is deemed so difficult to control. And this is because the nucleation rate, this is the expression for the nucleation rate in the classical nucleation theory. So we have the rate here, and the rate is just the number of crystals that nucleate per unit volume, per unit time. And so equal to some pre-exponential factor, and then there is exponential dependence of a free energy barrier. And here's the expression for the barrier. As you can see, this free energy barrier is a quadratic function of the driving force, the thermodynamic supersaturation. It's a cubic function of the surface free energy of the nucleus. Now, so these two factors, the surface free energy and the chemical potential difference, they enter the exponent. So basically, any way to control nucleation via these two parameters is extremely coarse. So nucleation either stops completely or proceed at undesired rate. So these are extremely coarse tools. But you can think of something else. Most of the time, especially in natural systems, you cannot control supersaturation because it's a function of concentration. And concentration is dictated by adjacent processes. Then you're left with one parameter, which is gamma. And gamma is the surface free energy. If you try to use modifiers to control gamma, these modifiers lead to a drop to the nucleus. If they are drop on the nucleus, they will lower gamma, which will make nucleation faster. So if you want to suppress nucleation by a modifier, you cannot do this according to classical nucleation theory. So you cannot do it for crystals that nucleate classically. And we're going to show an example where we use the fact that nucleation is non-classical and we design a means to control it. And so this example involves hematine. And hematine is part of the physiology of malaria parasite. So when the malaria parasite, during one of its life cycles, resides in the red blood cells, it consumes hemoglobin. And after it processes the hemoglobin amino acids to build its own proteins, it releases heme. So here is the heme. And then heme oxidizes to hematine because RN2 is unstable. And then hematine would be toxic and it will kill the parasite. But the parasite, of course, takes care of it by sequestering hematine into crystals. So you can see here the crystals in the digestive vacuole of the malaria parasite, which resides in a red blood cell. So in the last 20 years, it has been shown that inhibition of hematine crystallization, so removing this link in the process, is the most successful target for antimalarial drugs. There is a problem, and the problem is that the parasite is actually a very high-level organism, and it knows how to remove the drugs which control hematine crystallization. So we need to find new drugs, and to do this, we need to understand how hematine crystallization happens. And so that's what we did. We started to study how different drugs control crystallization. And there is chloroquine and quinine. And we found that they employ classical crystal growth inhibition mechanisms. So the drugs are dropped on the crystal surface. And if a step tries to penetrate between the two stoppers, it attains curvature. Curvature lowers the supersaturation and the step slows down. Then these other drugs, amodiaquine and methylquine, they employ different mechanisms. They attach to the kinks. The kinks are the only sites where a hematine molecule from the solution may attach to the crystal. And so by blocking the kinks, these drugs stop crystallization. Now, there are these new drugs which have been active only in the last 20 years, and the parasite is just beginning to develop resistance to them. These are the artemisinin class drugs. We found out that they are not active. This was our first work in 2017. They are not active in blocking crystallization. But then a few years later, we discovered that if these drugs form a complex with hematine, such as this one, which we call HART or HRs, so this heme artemisinin adduct actually works by blocking the kinks. And then we also found something very interesting. When we applied HRs to a hematine solution, to a growing hematine crystal, we found that at the same concentration of hematine, we see abundant nucleation of small crystallites that land on the surface of a brick crystal. This was very intriguing because it suggested that this drug can affect nucleation. And so we decided to see what other drugs would do. And for this, we use not AFM. It's a very impressive, but a little bit slow technique. We use dynamic light scattering. And you can see that if you're in a supersaturated solution of hematine, in the beginning of your observations, there is no signal, and then gradually a correlation function develops. And the shoulder of this correlation function gives us the sizes of the particles that form. And the amplitude of these correlation functions can be used to estimate the number of particles in the solution. So we applied this technique to supersaturated hematine solutions in the presence of these four drugs. And this is what we find. We find that chloroquine and methyloquine, they do not enhance, they do not suppress nucleation. So the evolution of the amplitude of the correlation function is identical to solutions, to supersaturated solution without any drug. But then here is the interesting part. HRs leads to nucleation. This is exactly in agreement with what we saw with AFM. And here's the most interesting part. Pyronaridine, this is this molecule here, it seems to suppress nucleation. And folks, this happens at two different supersaturations of hematine. So this is about 2 times supersaturated, this is about 3 times supersaturated. And you can see that at the higher supersaturation, nucleation happens much faster. So the suppression by pyronaridine was very impressive and also suggestive, because as we saw, classical nucleation theory cannot explain suppression of nucleation. So this suggests that we have a non-classical mechanism of nucleation. And yeah, so we confirmed this by electron microscopy. We went to Michael Sherman, an amazing electron microscopist in a university close to us, University of Texas Medical Branch. And he observed in hematine solutions particles which are of size about 100 nanometers. And the majority of these particles, if you record the diffraction pattern from them, they're amorphous. But then some of the particles, like this one here, they reveal a diffraction pattern, which appears to be a superposition of 2 diffraction patterns. So there is again the amorphous pattern, which suggests that the particle is still preserved. We have diffraction spots, which tell us that this particle holds crystals. Now, we can actually directly see crystals that have nucleated in this particle and then emerge from it. So hematine crystals are elongated like that. These observations tell us that, number one, that hematine forms clusters like this one. And then these clusters host the nucleation of crystals. So this is as expected for two-step crystal nucleation. And this thing was also observed by atomic force microscopy. So we saw formations which are irregularly shaped, which suggests that they are amorphous. And then in several instances, we were able to trace these formations over extended periods of time. And we saw that they evolved into faceted objects, which are probably crystals. Here is the other observation when we have amorphous formations on the substrate. There is no crystal here. And then we have a crystal which actually exhibits layers. So definitely, folks, we have two-step nucleation of crystals. And then the question is, so clearly, if we want to control nucleation, it would be a very good idea to control the population of the amorphous formations in which the crystals form. And then for this, we need to understand how these formations appear, what is their mechanism. And so we use oblique illumination microscopy. So the solution is held in a thin slide. Green laser is shown, and if you have particles, optical inhomogeneities, they will scatter light towards a microscope objective lens, and then these particles will diffuse. Yeah, my movie doesn't want to play. My computer is completely betraying me today. So you would see this spot participate in Brownian motion, and then we recorded the trajectory. There we go. So we recorded the trajectories of each of these particles. And then we did Stoch-Einstein's processing of each trajectory. And then we got the distribution of these particles by size. And you see that we have an average size, 80 nanometers, narrow size distribution. And the shape suggests that these particles are not the result of open-ended aggregation, such as a phase transition. And these particles exhibit other very interesting properties. So their size does not depend on time, so it's steady in time. Very importantly, it also doesn't depend on the driving force for the concentration of hematine. So they are clearly not one of those standard phases. They cannot be crystals because the solution is undersaturated, but they're also dense liquids because dense liquids would be stable and they would continue to grow in time, and they'd be bigger if you have bigger supersaturation. However, these behaviors correspond to mesoscopic solidly rich clusters, which we have studied in solutions of very, very different objects, proteins, small molecules. And we have actually worked with Bas Lubchenco And he has developed a rationale for this. So the idea is that your solids need to have a tendency to form transient dimers. And then when these dimers form, they create a gradient of the monomers, and the monomers diffuse towards the center of the gradient. And then there is this diffusion reaction dynamics, which leads to clusters, and the size actually doesn't depend on the concentration. It only depends on the diffusivity of these transient dimers. And now the clusters are, we know that they require transient dimers. The question is, can we affect the cluster population by the drugs? And we can see that chloroquine and methylquine affect the cluster population. This exactly correlates with their lack of effects on the crystal nucleation. Then look at H pyronaridine, and we see that HRs very strongly increases the cluster population, whereas pyronaridine very strongly suppresses the cluster population. And again, this is in accord with what these two drugs do to crystal nucleation. So pyronaridine suppresses crystal nucleation, HRs accelerates crystal nucleation very significantly. So we have a correlation. It seems that we have a control mechanism, but then the question is why? What do these drugs do to enhance or suppress nucleation? And the first thing that we tested is whether the drugs form complexes with hematine. And this is chloroquine. And these are the spectra of hematine titrated with chloroquine. So the concentration of chloroquine increases in this direction here and in this direction here. So you can see that some peaks are enhanced, other peaks are suppressed. So this clearly tells us that there is a complexation between hematine and chloroquine. And we can actually monitor the intensity of the peaks, and we can determine the equilibrium constant for this complexation and even the stoichiometry of the complex. And methylquine does exactly the same. So this is the change of absorbance in the presence of methylquine and compared to the initial absorbance. And clearly we also have a methylquine and hematine. So what this experiment shows, folks, is that chloroquine, methylquine form complexes. These complexes very strongly reduce the concentrations of the drug at the conditions where they are applied. So these are the purple areas in this diagram. And then these complexes passivate chloroquine and methylquine, and that's why chloroquine and methylquine do not affect the clusters and the crystal nucleation. Excellent. Let us now look what happens to HRs. So you can see here that HRs does not form complexes with hematine. So these are the spectra of hematine in the presence of HRs. They're exactly additive to the spectra of hematine and the spectrum of the drug. And then this curve here is horizontal. So this dependence of the absorbance compared to the absorbance with accounting for dilution, it's horizontal, irrespective of the titration, which tells us that there is no complex. So here is the hypothesis that we formulate. HRs contains four carboxyl groups. If these carboxyl groups would be charged, if they would be negative, they would repel hematine. So here is hematine, which also contains carboxyl group. But then there is a problem because our solution is mostly octanol. And so in octanol, these charges would be unstable. So probably those groups would be protonated and neutral. So to answer this question, we looked at the composition of the solvent for which hematine crystals grow. And it's important that this solvent is not pure octanol. It is octanol, which contains 5% water. This is the solubility of water in octanol. And the reason that we use such solvent is to mimic the lipid environment in the digestive vacuole. So this lipid environment is in contact with the purely aqueous environment in which process hemoglobin operate. So clearly, this lipid environment contains dissolved water, and so does octanol. And Lakshman Verma, helped by Jeremy Palmer, Jeremy is my colleague at UOH, Lakshman is a student that we co-advised. He carried out all atom MD simulations, and these are the results. So the blue balls are the oxygens of water, and you can see that they definitely form clusters. And this is the distribution of the clusters by their size. So about 50% of the water is held in clusters with a size at least 2. The average size is about 5. So how do these water clusters help us? Well, definitely these water clusters will concentrate on the hematine, on the carboxyl groups and stabilize them. And then the Coulomb repulsion outside of the water clusters will proceed through octanol, which has very low dielectric constant, so it will be long ranged. And so the Coulomb repulsion will increase the chemical potential of hematine and will drive hematine into the clusters. And the last one is pyronaridine, which suppresses nucleation. So pyridine also forms no complexes with hematine. And pyronaridine has two peculiarities. It has two triple amide groups, which are very basic, and they can be protonated and charged, positively charged. And then it contains this large flat entity, which increases very strongly its polarizability. So we suspect that these properties of pyronaridine make it interact with the hematine dimers, suppress the dimers, and in this way suppress cluster formation and nucleation of crystals. So I don't think I have time for this summary, but let me briefly tell you that the most important conclusion is that we have found a way in which antimalarials suppress the formation of clusters, which host the nucleation of hematine crystals. And these clusters exist owing to the reaction diffusion dynamics of transient hematine dimers. And then all these, so we found a way, we found the mechanisms by which the drugs affect the formation of dimers, and in this way, the formation of crystals. And so we present a rationale for control of crystal nucleation, which is one of the holy grails of material science. So thank you very much for your attention, and I'll be happy to answer your questions.
Thank you, Peter, for this interesting lecture. And so there is time for questions. Peter, I have a very general question for you. You, as an expert of nucleation of crystals, well, you showed us the famous slide with all the possible pathways. Crystallization is everywhere in nature. So do you think that there is a dominant pathway somewhere in nature? And if so, what pathway?
No,
do you think?
there is no dominant pathway. Actually, we'll keep discovering new non-classical pathways. And then you cannot even say that there is a dominant pathway for each individual system, because those pathways will depend on the conditions. So the same molecule, one and the same molecule in one solvent may choose one pathway, and in another solvent may choose a completely different pathway. So, you know, I want to say that non-classical nucleation is like an unhappy family. You know, Tolstoy said that all happy families are alike, and every unhappy family is unhappy in its own way. So I think that, yeah, all classical mechanisms are pretty much the same, but then all non-classical pathways are individual for the system that engages in them.
Thank you. Any other questions? I don't see any raised hands.
Well, this talk is the odd one out. There is no other talks on crystallization or nucleation.
So, Peter, thank you once again for this interesting talk.
Thank you very much.